the next presentation, which is by Jue Chen from the Rockefeller University and the Howard Hughes Medical Institute. And I think is Jue Chen here, or we're actually doing I think this is it's a presentation that's ready to go. So the title is ABC Transporters, Molecular Pumps Powered by ATP.
First, I'd like to thank the organizers for inviting me. It's wonderful to be part of this celebration. Our lab studied a group of molecules called ABC transporters. ABC, the name comes from the presence of two ATP binding cassettes, also called nucleotide binding domains in the molecule. Some ABC transporters are importers, bringing nutrients into the cell. Others are exporters, exporting a variety of substrates, as small as ions and as large as proteins outside the cell. If you take a bacterial cell you grow in the lab, usually they have about 80 different ABC transporters. We humans have 48 different transporters. So today, I like to give two examples. The first example will be a typical ABC exporter, and I like to use this example, MRP1, to illustrate the principles how ATP binding and hydrolysis are coupled to substrate translocation. The second example will be an unusual ABC transporter, which is evolved into an ion channel. So instead of actively pump substrate against its gradient, CFTR is an ion channel that will let ions run down its chemical electrogradient. So let's first look at MRP1. MRP1 is a single polypeptide containing three transmembrane domains, TMD0, TMD1, and the TMD2. And the two nucleotide binding domains called NBD1 and NBD2. The function of the N-terminal TMD0 is not clear, and the rest of the molecules form the core transporter, similar to many other ABC transporters in nature. MRP1 is widely expressed in many different tissues, and its job is to protect us from toxic molecules. However, in several cancers, especially neuroblastoma, MRP1 was found to be related to multidrug resistance. MRP1 transport a large array of different substrates, but they have a common motif, usually has a hydrophobic region and a negative charged region. And the subject of today's talk is called the leukotriene T4. It's a molecule synthesized and secreted by immune cells during inflammation. The structure of MRP1 in multiple conformations were determined almost single-handedly by Zach Johnson, a former postdoc in my lab. This is the structure of MRP1 determined in the absence of ATP and the substrate. So we often call it the resting state. In this conformation, you can see the TMD0, which colored red here, is loosely attached to the rest of the transporter. TMD1 and 2 together form a translocation pathway that is connected to inside the cell. The two nucleotide binding domains, NBD1 and NBD2, are far apart from each other. So we call this an inward-facing conformation, coupled with separated NBDs. Residues that are known to bind leukotriene C4, several of them are highlighted here, are located in the center of the membrane, contribute from two halves of the transporter, both from TMD1 and the TMD2. So binding leukotriene C4 to the transporter in the absence of ATP will cause the two halves of the NBDs to rotate towards each other pin them together. So if we compare the two conformations, let's first focus on the substrate binding side. You can see the residues come close together, and together they form a very high affinity binding site to the substrate. This conformational change is propagated about 50 angstroms away into the nucleotide binding domains and causing the two nucleotide binding domains to come closer to each other. So I'm going back to the apo form and the leukotriene binding site conformation. So in this conformation, leukotriene C4 binds at a KD about 300 nanomolar. So how does this high affinity binding site release its substrate to the other side of the membrane? Watch what happens when we put ATP in the sample. The structure determined in the presence of ATP, immediately you can see the binding site is restructured. Residues forming a leukotriene C4 binding now are pulled apart. This conformational change will lower its affinity for leukotriene C4. At the same time, the transmembrane domains are rotated. Now the binding site is open to outside the cell, and this is exactly the conformation leukotriene C4 will be released into the extracellular media. So in fact, the structure was determined at high concentrations of leukotriene C4, but we don't see any density at the binding site, indicating this site has very low affinity to avoid leukotriene C4 come back, to make sure that transporting is unidirectional. So what happens at the nucleotide binding domain is now you can see, compared to the ATP-free conformation, adding of the ATP stabilized the two NBDs into a closed dimer conformation with two ATPs sandwiched at the very interface. So in this conformation, ATP is positioned here, what I will show outline here, sandwiched in between the two halves, two NBDs with all the catalytic residues positioned to hydrolyze ATP. The reason this molecule is not hydrolyzed is because we made a mutation at the catalytic side, so it will only be trapped in the ATP-bound conformation. Putting the three structures together, we can come up with a working model how ATP hydrolysis is coupled to substrate translocation. So in the absence of substrate and ATP, MRP1 resting in the inward-facing conformation with its binding site connected to the cytosol. Binding of the substrate, such as leukotriene C4, will bring the two halves of the molecules closer to each other, prime the nucleotide binding domains for ATP binding. Subsequent ATP binding will stabilize NBD dimerized conformation. At the same time, rotate the transmembrane domains to alternate the access to the binding site to the outside the cell. Also, because of the conformational change at the binding site, the affinity for leukotriene C4 is much lower now. It's ready to release the substrate. So here, what we demonstrate is ATP binding instead of hydrolysis was sufficient to release the substrate from the transporter. And the function of the hydrolysis is to reset the transporter into an NBD separated inward facing conformation. So MRP1 is a drug exporter from eukaryotic cell. So next, I'd like to show you very quickly what we learned from a very different ABC transporter. And this is a bacteria imported responsible for bringing sugar molecules into bacteria. And it also has two transmembrane domains, two nucleotide binding domains, with accessory substrate binding domain. In this case is the maltose binding protein. So the structure of maltose transporter was determined also in three different conformations. At the beginning, at the first glance, you'll say they look so different from MRP1. But if you look carefully, several general principles we learned from MRP1 can also be applied here. For example, in the absence of substrate and ATP, maltose transporter also rests in an inward-facing state with its two nucleotide binding domains separate from each other and the maltose binding side facing inside. But it's an importer, which means the substrate has to be delivered from outside the cell. And binding of the substrate is delivered through the binding protein, which is shown here. So binding of the binding protein to maltose transporter also stabilize the conformation where the two nucleotide binding domains are much closer to each other. And in this conformation, ATP binding will stabilize the NBD dimer, now reorientate the binding site towards the extracellular, in this case, is a paraplasmic binding protein. And because it's an importer in this conformation, the substrate is delivered from the binding protein into the transporter, into the membrane. Again, ATP hydrolysis reset the transporter into an inward-facing conformation by separating the two NBDs. So when we put the two systems together, something very beautiful I find. Although these two transporters are from very different organisms and they also move substrate at opposite directions, several principles such as substrate binding primes the transporter for ATP binding by bringing the NBDs together. But functionally, we can determine ATP hydrolysis turnover rate is accelerated through the presence of substrate. And that's because in ATP hydrolysis occurs in the outward-facing state. By prime the transporter bringing two NBDs together, presence of substrate accelerate this process. Also, through the same coupled conformational change, will the closed NBD dimer to coupled to the outward-facing state, the exporter will release the substrate and the importer will receive the substrate. So in this half of the transporter cycle, ATP functions like a typical ligand, stabilize the particular conformation, which is the outward-facing conformation for both transporters. But at the very same conformation, it also positions itself for hydrolysis. So in the NBD dimerized state, ATP will be hydrolyzed and the ligand is now changed to ADP and the energy will be used to reset the transporter into an inward-facing conformation. The same is true for both exporters and importers. So next, I'd like to discuss a very special case. That's a protein called the CFTR. The structures I'll present it today and functional data are done by an amazing team, postdoc Jue Chen and the graduate student Fengyu Liu. So when CFTR, the gene, was cloned in 1989, it came out as a surprise, because at that time, it was already known that the disease, cystic fibrosis, is related to abnormality in iron permeation. So it was assumed that the very protein responsible for the disease is the ion channel. But when CFTR gene was cloned, it looks like a transporter, very similar to MRP1, as you'll see. It has two transmembrane domains and the two nucleotide binding domain. What's unique in CFTR is the presence of the regulatory domain, which is colored in red here. So the protein was named Cystic Fibrosis Transmembrane Conductance Regulator, with the idea that this is a transporter, might transport a particular regulatory molecule that will in turn regulate the ion conduction of an ion channel. It turns out there is no other ion channel. CFTR itself is the ion channel. So this is a patch of membrane with a single CFTR reconstitute, and the time is shown as X, and the current is shown as Y-axis. And this part of the record is quiet, means ATP binding alone is insufficient to activate or open the CFTR channel. Treatment with pKa will phosphorylate the protein, particularly the R domain, and only after phosphorylation we can see the channel starts to open and the molecules flip through between the open conformation and the channel closed conformation. Phosphorylation not only regulates the channel conductance, it also regulates the ATPase activity. And here is the ATP turnover rate as a concentration of ATP. And as you'll see, only the pKa treated CFTR can hydrolyze ATP efficiently. So the question is how? How does the regulatory domain regulate the phosphorylation, regulates the channel activity and ATP hydrolysis? To address that question, let's first look at the structure of CFTR corresponding to this part of the record where there is no ATP and no phosphorylation. And this structure must remind you of MRP1. It also has two transmembrane domains and the two nucleotide binding domains that are separate from each other. What's different is the presence of R domain that's colored in red here. The ion conduction pathway previously was mapped extensively through mutagenesis. It's outlined here as a gray mesh, and it's located right in between the two half of the molecules. A similar location where leukotriene C4 would bind to MRP1. So next, let's look at the structure of a phosphorylated CFTR in the presence of ATP, which corresponding to this part of the record. And this structure also reminds you about the outward-facing conformation of MRP1 in a way. The two nucleotide binding domains form a closed dimer. The two NBD transmembrane domains coming much closer to each other, and the R domain, colored in red here, is in a completely different location. So when we compare the two structures and just focus on what happens to the R domain, here in the dephosphorylated ATP-free conformation, the R domain is tucked in between the two nucleotide binding domains and the transmembrane helices 9 and 10. And in this position, the two nucleotide binding domains will not be able to come closer to each other. After phosphorylation and in the presence of ATP, the NBDs form a closed dimer, and the intracellular opening between helices 9 and 10 is completely closed. Now our domain is being displaced on the surface of this molecule. So putting these two structures next to each other, we come up with a hypothesis to explain why R domain phosphorylation is key in regulation. So in the absence of phosphorylation, the R domain functions as an autoinhibitor. It jams in between the two halves of the molecules, preventing the NBD form a closed dimer and the two transmembrane domains to come closer to each other. Upon phosphorylation, usually many different phosphorylation sites has to be phosphorylated on R domain. Now it's being displaced or stabilized outside the cavity, and only in this conformation the inhibition is lifted and the NBDs can come together and allow the channel to open. So next I want to answer the question, why is CFTR an ion channel, not an active transporter? Many of us know ABC transporters and and other active transporters function through what we call the alternating access mechanism. So it has two basic conformations, an inward-facing conformation and outward-facing conformation. For ABC transporters, the inward-facing conformation is coupled to NBD-separated structure, and the outward-facing conformation is stabilized by a closed NBD with two ATPs bound at its interface. We now see that CFTR also alternates between those two basic conformations. But when I compare the structure of the NBD closed CFTR with that of MRP1, we can see a difference. So the structure of MRP1 in the outward-facing conformation has what we call the intracellular gate closed, which is highlighted here. And you can see the helices 9 and 11 are in close contact with each other. And this is this part of the structure. So the transporter is only open to one side of the membrane. But the very same region in CFTR is cracked open. The corresponding helices 4 and 6 are open, have separated from each other, and the positively charged residues line this opening, attracting chloride or other anion ions into the translocation pathway. So in the ABC transporter field, we like to call CFTR the broken transporter. Now we know exactly where it's broken. The intracellular gate is broken in the NBD dimerized phosphorylated state. And that's how the channel, it can have an open passage across both sides of the membrane. So to summarize today's talk, I first showed you examples of a typical ABC transporter and described how substrate binding primes the transporter for ATP hydrolysis, and ATP binding stabilize the outward-facing conformation to either release the substrate outside the cell or receive the substrate into the membrane. And ATP hydrolysis function to reset the transporter into an inward-facing conformation to restart the transporter cycle. Lately, then I presented two structures of CFTR and focus on why R-domain phosphorylation regulates channel activity, also why CFTR functions as ion channel. That's because the intracellular gate is broken, it is cracked open in the NBD dimerized form to allow the transmembrane pathway open to both sides of the membrane. I like to thank many wonderful people contribute to the work, and particularly Jue Chen and Feng Yu. Now the CF project is continued by another postdoc and the student team, Carol and Jasper. MRP1 project was mostly done by Zach with help from Jasper and Holland, two graduate students in the lab, and many wonderful collaborators contributed to different parts of the work, which I didn't have time to talk about. Thank you very much for your time.
Okay, great. Well, thank you, Chenju. Actually, because of the logistics, I missed the last 1 minute of the presentation, the way it's set up. But what I heard was very, very exciting and very relevant. So I have a few questions in the chat here. One comes from Robert Jernigan. Do you think it may be possible that the ATP, and this is related to the ABC transporter, the ATP hydrolysis exerts a force to cause the conformational change? I believe that many such changes are being attributed to binding rather than the dynamics of the hydrolysis itself. So what do you think of that, Jue Chen?
So absolutely, ATP binding and hydrolysis each have a different role in the transporter cycle. The energy from binding is used to stabilize the conformation where the NBD is dimerized. Then hydrolysis is essential to reset the transporter to separate the two NBDs so the transporter can go back to its resting state. So we know without hydrolysis in many transporters, if we mutate the catalytic residues, the transporter will stuck in the outward-facing NBD dimerized state. So hydrolysis contributes to the conformational change to separate the two NBDs.
Thank you. Okay. So, and these questions are going to be available to Jue Chen, and you can email back and forth. So Doreen Mathias asks, what is the role, and again, this is MRP1, what is the role of the red extra transmembrane domain in MRP1?
We don't know. Yeah, it's a very good question. So for MRP1, we have no clue what the additional transmembrane domain does. In a homologous protein called the TAP, which is a transporter associated with peptide presentation, the similar domain is involved in interacting with another protein called the taposin. Also in KATPase, a homologous domain is involved in, again, interacting with another molecule. So it's possible TMD0 in MRP1 is interacting with potentially another partner. We just don't know yet. We're very interested in identifying that.
So now this question is regarding CFTR. It's from Rumas Brahma. Is the breaking in the CFTR inducing conformational change in the R domain?
I'm sorry, I don't understand the question.
Breaking in the CFTR, inducing the conformational change. I'm not sure either. Maybe I'll move on to another question then in terms of time. From Carol Burka, what is the function of the LO or L0 domain in MRP1 transporter? Does it move during the transport cycle? No, we didn't see any movement of this region in the transport cycle. Kathleen Agertes asks, can you please comment on the CFTR flickered closed conformation and its role in channel function?
Yeah, great question. CFTR is flickering even in the open state. What we know is in a very fast time scale, so it's probably likely to be a local conformational change. We have different ideas what local conformational change may cause it, but we don't have data supporting those hypotheses yet. We'll study that further.
And one more, there's a few more in the chat, you can see them later. This is from Stephanie Rosario Garrido. Did you use liposomes to test the transport activity of multidrug resistance transporter 1? No, we haven't been able to do that. And I just want to ask us a general question, which is, in these membrane protein structure studies, how can you be sure that the structure that you see in a reconstituted system is what actually happens in the cell, in the biology? To what degree maybe reconstitution and the membrane environments that you're able to use make the structure not exactly what it is? We can never be sure. We can only test. We see what we see. There are lots of, when we try to correlate what we see structurally to what we learned through function, and we design mutations, we make predictions and we test. Of course, it would be much better to have people ask the questions themselves, because then they're properly asked and clarified. But thank you very much for a beautiful presentation and really cool work. Thank you.
Thank you.